Okay, so we ended up talking about glycolysis. So this is the lysis of glucose, right? This occurs in the cytosol without oxygen and yields um, two molecules net of ATP. It also yields two molecules of NADH, which has lots of energy, and two molecules of pyruvate, which has energy to be extracted, at least if oxygen is present. So now we're going to see how we can get to that energy when oxygen is present. This involves the second step of cellular respiration, which is the Krebs cycle or the citric acid cycle. So those are interchangeable in terms of what those mean. So a few things must happen. First, the pyruvate that is produced in glycolysis um, first has to go from the cytosol into the mitochondria. So the transport of pyruvate into the mitochondria only occurs when oxygen is present. Now we will see that oxygen isn't needed in the citric acid cycle. We actually don't use oxygen there. The only place oxygen comes in is in oxidative phosphorylation in the last step um, of cellular respiration. But there are mechanisms in place so that you don't begin the process of breaking down this pyruvate unless the cell knows that oxygen will be there by the end of the process. So pyruvate goes into the mitochondria, and we'll look at the structure of the mitochondria a little bit later. And the first step to prepare this pyruvate for the citric acid cycle is is what's called pyruvate dehydrogenation or decarboxylation. And you can actually look at this pyruvate and see that there is a nice little CO2, so that would be a carbon dioxide unit that's ready to go off of this pyruvate. And so in this process, you chop off that carbon dioxide. This actually forms the first of three processes that remove carbon dioxide as a waste product. And then you take the remaining group, the CH3 um, CO, and you link it to an enzyme called coenzyme A. Now we will see that every time we remove a carbon dioxide, we generate an NADH. And remember, there's a lot of energy in those NADHs. So this process to prep the pyruvate to go into the citric acid cycle translocates pyruvate into the mitochondria, removes the carbon dioxide from that pyruvate to form an acetyl group, links that acetyl group to the enzyme coenzyme A, and forms a reduced electron carrier NADH in the process. So this is the first time that we've actually lost a carbon from glucose. And I'll just remind you from glycolysis, you have two pyruvates that are formed from one glucose molecule. So this process, um, occurs twice for each glucose molecule. And so here we would have two molecules of acetyl-CoA produced, two molecules of NADH, and 2 of the 6 molecules of CO2 come out here, right? Okay, so now we have the acetyl-CoA. So this is just another look at this. You have this glucose, it goes to pyruvate, it gets converted to an acetyl-CoA group to prepare it to go into the citric acid cycle. Now, one of the things that we learn in biochemistry is that these acetyl-CoA groups here, um, almost every digestive kind of breakdown that we do ends up making acetyl-CoA. And so we converge all of our catabolic activity on these acetyl groups to feed into the Krebs cycle. And again, this does take place in the matrix of the mitochondria. And so this is a Krebs cycle. This is kind of a broad overview, and then we'll go into a little bit more depth. But essentially, you start again counting carbons here. You start with the 4 carbon structure oxaloacetate. You then add in 2 carbons from the acetyl-CoA group to form a 6 carbon structure citrate, and then we're going to go through 2 rounds of decarboxylation. So we're going to remove carbon dioxide twice. That leaves you back to a 4 carbon structure. Every time we remove a carbon dioxide, we create a NADH, and then we're going to oxidize that structure back to oxaloacetate so that it can take a pick up another acetyl group. It's a cycle because you end up right where you start. And again, because you form 2 acetyl groups for every 1 glucose, you run this cycle twice to get the total amount of energy that you get out from 1 glucose molecule. So here in the citric acid cycle, you produce a little bit of ATP, 2 molecules of ATP, but you end up producing a lot of NADH. NADH there, there. Actually, you get 2 molecules of NADH there. And then another electron carrier, FADH2. And so you end up making 6 molecules of NADH and 2 molecules of FADH2. And remember, there is lots of energy in these reduced electron carriers. So that's where most of the payoff is going to be. Up until this point, we have only generated a net of 4 ATP for all the work that we've done, right? That's not a lot of energy for what we've done. So this is a breakdown of what is going on. You start the cycle with this acetyl-CoA coming in. Um, the coenzyme A is chopped off, and then that goes back to pick up another acetyl group. And for cycles, they're in what's called a dynamic steady state. And so this is a condition in which the atoms within the intermediates are changing, but the concentrations of those intermediates don't change. And so even though we're throwing stuff into the system, the steady state concentrations of those intermediates stay the same. And at the end of this, we're going to get CO2. So this is where all of the remaining CO2 gets processed. So that would be 4 of 6 molecules. You get a bunch of reduced electron carriers and a little bit of ATP. Well, that doesn't show up well, but this should be an animation in the PowerPoints. Um, but this is the structure of the mitochondria. Mitochondria is a dual membrane system, so it has a permeable outer membrane. So it has these huge holes in it um, that allow for basically anything to go through. and then a semi-permeable inner membrane. And if we remember our talks about lipid bilayers, they look like that, right? And they have what? Hydrophilic heads and hydrophobic tails, right? And so a lipid bilayer that's semi-permeable allows for the flow of hydrophobic residues or hydrophobic um, compounds, um, but blocks any charged molecules. In particular, for this case, the proton, H plus, would be stuck on one side of the inner membrane or not. And so we're going to see in oxidative phosphorylation how profound a deal that is. And so the processes of the citric acid cycle occur within the matrix on the inside of the mitochondria. And then everything is stuck there unless they have some way to get through the inner membrane of the mitochondria. I'll say too that the inner membrane of the mitochondria has these folds. If you remember from the previous chapter, those are called cristae. And what this does is help increase the surface area of the inner membrane of the mitochondria. It turns out that the electron transport chain is on the membrane of that mitochondria. And so by increasing the surface area, you allow for more of these chains to be placed, which lets you generate more ATP per unit volume. Okay, so um, this This is talking about the pyruvate oxidation or decarboxylation step that we already talked about. Again, you have this pyruvate molecule, one, two, three carbons, and then you remove a carbon dioxide, this guy, you generate an NADH, and then you create the remaining two carbons, one, two, and attach it to this coenzyme A. This coenzyme A is basically a pedestal for the acetyl group to come in. And that goes to citric acid cycle. And this, my friends, is the citric acid cycle. Now, for our purposes in General Biology 111, I'm not going to ask that you memorize structures and all of that sort of thing, although you certainly will do so in biochemistry. But again, let's just go over the big point. And as always, you should be doing two things, counting carbons and um, following the electrons, right? And so here you start and get something bright with oxoacetate. And this is one, two, three, four carbons there. You add the two carbon acetyl group and you form citrate. And so this is one, two, three, four, five, six carbons. So you form a six carbon citrate by adding two carbons from acetyl CoA, which is, I don't know, up here someplace. I don't know what happened to it. From there, well, if you can kind of look, it might be a little bit easier to see here. So the first state here is to isomerize the citrate, and that's just to move this alcohol group here. It turns out that there's some good chemistry that makes that occur, but we won't go to it here. But you still have, since it's an isomerization reaction, you still have the same um, uh, uh, formula. So here, one, two, three, four, five, six carbon dioxides. And if you'll note, there are one, two, at least three places where you can lose the carbon dioxide. Now we're going to go three rounds of decarboxylation. So really, and I tell this to the biochemistry students, you know, there's only three places that you can remove these carbon dioxides, and so you can almost guess and get the right one without even knowing much organic chemistry in order to get there. But for our purposes, we have this six carbon structure. We're going to undergo a round of decarboxylation. So we're going to lose a carbon dioxide. Whenever we do that, we generate an NADH, a reduced electron carrier. So now we have one, two, three, four, the five carbon alpha ketoglutarate. We're going to go through another round of decarboxylation. So boom, lose a carbon dioxide, generate a reduced electron carrier NADH, and then you're left with a one, two, three, four carbon succinyl group that is reattached to a coenzyme A. So this is the same coenzyme A that we saw in the beginning of the cycle. I mean, not literally the same one, but the same structure of molecule. And so now we're back to the four carbons that we started with up here, but there's still a lot of energy embedded in this compound. And so we're going to remove that coenzyme A, in the process make a little ATP via GTP. You're going to form a four carbon succinate group, and then we're just going to oxidize that succinate group to form fumarate, then malate, then back to oxaloacetate. And so here, once we get back to that four carbons, we're just pulling off the energy in the form of FADH2 and NADH to get a little bit more bang out of this process. And there we have it. We just got through the citric acid cycle. The result here is that we have generated all of the carbon. So we've lost all the carbon from that six carbon glucose. Four carbon dioxides are generated here, two in pyruvate oxidation. Um, and we've generated a bunch of reduced electron carriers. And if we look at our Gibbs free energy here, we can see this is where we were with glycolysis. So we started with glucose, which is up here at just normalized to zero. We pulled out some of the energy, the potential energy of that glucose to form pyruvate. Pyruvate is of lower energy. Stop that. Is of lower energy than that glucose. And then in the citric acid cycle, we pulled out even more energy. Oxaloacetate is at significantly lower energy than this pyruvate, and we have captured as much as possible in the form of NADH, FADH2, and uh, well, this and ATP. So each glucose here, so this is wrong. I'm doing this is an old slide. It yields six carbon dioxides, 10 NADH, 2, 4, 6, 8, 10, because two from glycolysis, two FADH2, and four ATP. Um, and so that's the second step, the citric acid cycle. Maybe I'll stop here and then just do oxidative phosphorylation. Yeah, I guess I'll do it that way. Um, so this is the second step of the of cellular respiration, looking at pyruvate oxidation and the citric acid cycle. Again, all of this, now we've completely broken down the glucose. Oops. But for all that work, I mean, 8 steps in the citric acid cycle and 10 steps in glycolysis, plus the pyruvate oxidation, which is like a three enzyme process. Um, and we've only formed 4 ATP net. All right. So that can be used by the cell. So thus far, we've done a lot of work, but we haven't gotten a lot of payoff out of it. And so what we do have that the cell doesn't directly use are all of these reduced electron carriers, these 12 reduced electron carriers. And so we have to figure out how to get access to that. And that's what happens in oxidative phosphorylation. that we'll talk about next. See you then.